Welcome to the PubMed Daily Review for April 13th, 2026. Today we scanned 2007 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Cancer Biomarkers and Treatment Efficacy. These papers explore how biomarkers inform cancer diagnosis, prognosis, and treatment effectiveness. This cluster covers 245 papers. Representative work includes identification of hidden cachexia subgroup in PD-L1, high NSCLC, comparative analysis of the AWGC versus Farron criteria, ependymoma group-specific blood-brain barrier differences uncovered by a multi-omics approach, and correlation of ECAD-herin, Vimentin, CD206, programmed cell death receptor 1, and programmed cell death ligand 1 expressions with clinicopathological factors and prognosis in oral squamous cell carcinoma. Trend 2 of 5, Neurological Aging and Brain Health. These papers explore neurological aging and brain health through diverse methodologies, from clinical studies to computational modeling. This cluster covers 485 papers. Representative work includes treatment-modifying effects of frailty on stroke reperfusion therapy outcomes, a systematic review and meta-analysis, frequent in dementia, deadliest without it, delirium and mortality in hospitalized older adults, and the role of dopamine decline, astrocyte reactivity, and cerebral small vessel disease in cognitive aging. Trend 3 of 5, Cardiovascular Disease and Biomarkers. These papers explore biomarkers and integrated approaches for understanding, predicting, and managing cardiovascular and related diseases. This cluster covers 437 papers. Representative work includes an integrated biobank in the Swedish heart failure registry, clinomics, proteomics, transcriptomics, and genomics. Endothelial progenitor cell numbers vary in newborns and adults. Anti-GM-CSF-alpha, CD116, antibody as a novel marker for EPC enumeration, and demographics and mortality trends of hypertensive heart failure in the United States, 2006 to 2020. Insights from the CDC Wonder Database. Trend 4 of 5, Health Promotion and Care Access. These papers explore improving health outcomes through better access to care, services, and health promotion strategies. This cluster covers 313 papers. Representative work includes assessing the quality of lactation accommodation policies in California's higher education system, cervical cancer prevention behaviors and determinants among indigenous women in rural Nepal, and health promotion at the crossroads, putting civil society in the driver's seat. Trend 5 of 5, Microbial and Material Science Innovations. This trend explores how biological and material science advancements intersect to solve complex problems. This cluster covers 527 papers. Representative work includes synergistic activity of cefepime/enmetazobactam with meropenem, piperacillin/tazobactam, cefiderocol, and phosphomycin against Klebsiella pneumoniae carrying black APC allelic variants, tick-borne pathogens in Ixodes ricinus, acari, Ixodidae, and Ixodes inopinatus xi, ricinus hybrids in Central Europe. No link to bird diversity or abundance, and electrically responsive multi-layer soft actuators using a solvent-free high dielectric permittivity. Polysiloxane Inc. Up next, our first paper out of 5, Precise Gene Editing of Pathogenic Lamin A Mutations Corrects Cardiac Disease by Serd M. Caravaya and Eric N. Olson from A Department of Molecular Biology, Hammond Center for Regenerative Science and Medicine, University of Texas Southwestern Medical Center, Dallas, Texas, 75390. Let's break down its key contributions. Laminopathies, caused by mutations in the Lamin A gene, are severe genetic disorders affecting the heart and muscles. Unfortunately, current treatments are limited. This research investigates whether precise gene editing can correct these disease-causing mutations and restore normal function. The researchers developed precise base editing tools, ABE and CBE, to target specific Lamin A mutations. They tested these tools in human heart cells derived from stem cells and in mouse models carrying these mutations. The goal was to see if correcting the gene could reverse the disease-causing effects observed in these models. The results were highly encouraging. Gene editing successfully corrected the cellular problems in heart cells carrying the mutations. Furthermore, when delivered to mice, the base editing approach prevented the severe cardiac and muscle disease symptoms and significantly improved their lifespan. This demonstrates a powerful therapeutic potential. This study shows that precise gene editing is a viable and effective approach for tackling devastating genetic heart and muscle diseases caused by Lamin A mutations. It opens a new avenue for developing therapies for laminopathies. While these findings are exciting, further studies will be crucial to confirm safety and efficacy before human trials can begin. Up next, our second paper out of 5. CRISPR activation screens map the genomic landscape of cancer glycome remodeling by John Daly and Simon Wisnowski from Faculty of Pharmaceutical Sciences, University of British Columbia, Vancouver, British Columbia V6T1Z3, Canada. Let's break down its key contributions. Many cancers have a way of hiding from our immune system by changing the sugars on their surface. These altered sugars, called glycans, can essentially tell immune cells to back off. This allows the cancer to grow undetected. While we know this happens, the specific genetic changes that cause it are still a bit of a mystery. Unraveling these genetic pathways is key to finding new ways to fight cancer, especially through immunotherapy. To tackle this question, the researchers used a powerful technique called CRISPR activation screening. This allowed them to systematically turn on genes in cancer cells and see which ones affected the expression of those immune-evading glycans. They performed multiple screens to get a broad picture. Their approach focused on understanding the genetic competition between enzymes that build up these specific glycan structures and also looked for other factors that might enhance this interaction. Finally, they connected their findings back to real-world cancer data. The screens revealed a fascinating competition between different enzyme pathways that control glycan structure. They found that cancers can remodel their surface by shifting this balance. Importantly, they identified new professional ligands that enhance the binding to immune-inhibiting Siglec receptors. They also showed that a known gene, CD24, isn't actually necessary for this interaction with Siglec-10. A particularly exciting finding was the identification of the sulfotransferase GL3ST4 as a potential driver of immune evasion in glioma cells. This study gives us an unprecedented look at the genetic landscape of how cancer cells change their sugars to hide from the immune system. The findings pinpoint specific genes and pathways that can be targeted for new immunotherapies. In particular, the identification of GL3ST4 opens up a potential new avenue for treating gliomas. This work lays the foundation for developing more effective treatments that leverage the body's own immune system. Up next, our third paper out of 5, Decoding the Glycan Shield, Immune Recognition and Response to the HIV-1 Envelope Trimer by Fatima Zarnawaz and Fikri Y. Afsi. Let's break down its key contributions. The HIV-1 envelope glycoprotein, or ENV, is the main target for antibodies that could neutralize the virus. But ENV is covered in a dense layer of sugar molecules, called glycans, which act like a shield, hiding the virus from our immune system. For decades, scientists have tried to create better ways to present ENV to the immune system to generate protective antibodies. This review delves into how these glycans not only hide ENV from B cells, but also play a role in how T cells recognize the virus, which is critical for developing a successful HIV vaccine. This review synthesizes current knowledge on the HIV-1 envelope glycoprotein, focusing on the complex role of its glycan shield. Researchers examined how the extensive glycosylation of ENV impacts both B cell recognition, which is key for antibody production, and T cell responses. The review analyzes how these glycans can both hide viral targets and, perhaps surprisingly, contribute to the processing of viral fragments that T cells can recognize, offering new insights into immune evasion and potential vaccine strategies. The study highlighted that the HIV-ENV trimer is covered by a remarkable shield of up to 90 N-linked glycosylation sites. This extensive glycosylation was found to effectively shield the underlying protein from recognition by B cells, a major hurdle for antibody-based vaccines. However, the research also revealed that these glycans can function as unconventional epitopes for CD4+ T cells, suggesting a more complex role in shaping the overall immune response. Essentially, glycosylation simultaneously constrains B-cell immunity while contributing to T-cell-mediated responses. This review underscores that understanding the intricate relationship between HIV-ENV, glycosylation, and immune recognition is paramount for developing an effective HIV vaccine. Future vaccine strategies must find ways to effectively bypass or penetrate this glycan shield to elicit strong, neutralizing antibody responses. Furthermore, exploring how to harness the T-cell epitopes presented by these glycans could be a key to boosting overall vaccine efficacy and achieving lasting protection against HIV-1 infection. Up next, our fourth paper out of 5. The efficacy and safety of ultrasound-guided nerve block in the treatment of cervical spondylotic radiculopathy, a systematic review and meta-analysis, response to letter, by Mingyu Deng and Jiai Quang from Graduate School, Guangxi University of Chinese Medicine, Nanning, Guangxi, People's Republic of China. Let's break down its key contributions. Cervical spondylotic radiculopathy can significantly impact quality of life. While various treatments exist, ultrasound-guided nerve blocks have emerged as a promising minimally invasive option. This meta-analysis sought to consolidate the evidence to understand their true efficacy and safety profile. This study is a systematic review and meta-analysis, pooling data from multiple primary studies. They included a total of 1,072 patients diagnosed with cervical spondylotic radiculopathy. The intervention was an ultrasound-guided nerve block compared against control groups. Key outcomes assessed included pain scores, functional status, and procedural success rates. The meta-analysis revealed significant improvements favoring the ultrasound-guided nerve block group. For the physical component summary, the mean difference was plus 3.50, 95% CI 1.26, 5.75. Similarly, the mental component summary showed a mean difference of plus 2.22, 95% CI 0.79, 3.64. These results indicate a clear benefit of the intervention over control. In conclusion, this meta-analysis supports the use of ultrasound-guided nerve blocks for cervical spondylotic radiculopathy, demonstrating significant improvements in patient-reported outcomes. However, the authors acknowledge limitations in the quality of evidence, particularly regarding risk of bias and heterogeneity. Future research should focus on addressing these limitations to provide more robust evidence. Up next, our fifth paper out of 5. Nirsevimab in high-risk infants in a respiratory syncytial virus prevention strategy by Dennis Sohr and Leonardo J. Basso from Industrial Engineering Department, Universidad de Chile, Santiago, Chile. Let's break down its key contributions. Respiratory syncytial virus, or RSV, is a significant threat to young children, especially those who are born prematurely or have underlying health conditions like congenital heart disease. While we have tools to protect healthy infants, understanding how well these protect our most vulnerable babies has been a key question. This study aimed to answer just that by looking at the real-world effectiveness of Nirsevimab in these high-risk infants. To investigate this, researchers in Chile conducted a case-control study using national health registries. They looked at infants who were hospitalized with RSV-related illness, comparing them to similar infants who were not hospitalized. The key intervention was the administration of a single dose of Nirsevimab, a long-acting antibody. The study focused on the 2024 RSV season and examined RSV-related hospitalizations as the primary outcome. The results were quite striking. Nirsevimab was associated with an impressive 84.3% reduction in RSV-related hospitalizations among all at-risk infants studied. This protection was particularly strong in infants with congenital heart disease, showing a 96.3% reduction. While the reduction for extremely preterm infants was lower at 65.9%, it still indicates a significant protective effect. Overall, these findings demonstrate Nirsevimab's effectiveness in preventing severe RSV outcomes in vulnerable populations. So, what does this mean for clinical practice? This study strongly suggests that a universal Nirsevimab immunization strategy can substantially reduce RSV hospitalizations in infants at higher risk of severe disease. While the protection was most pronounced in infants with congenital heart disease, it was still significant across the board. These findings provide compelling evidence to support a shift from targeted palivizumab prophylaxis to a broader Nirsevimab-based approach for RSV prevention in these vulnerable populations. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.